Gene therapy is an exciting new field that aims to treat diseases by directly modifying the genetic makeup of cells. This revolutionary approach has the potential to address the underlying causes of many diseases, offering hope for cures that were previously out of reach. The image on this slide depicts a futuristic lab setting, with a scientist working on a cutting-edge gene therapy machine surrounded by glowing DNA strands. This symbolizes the advanced, scientific nature of this technology. Gene therapy is still an emerging field, but the progress made so far is incredibly promising and could lead to transformative breakthroughs in medicine. Gene therapy is a powerful approach that involves modifying the genetic makeup of cells to treat or prevent diseases. The goal is to correct faulty genes that cause genetic disorders, which can lead to improved health outcomes for patients. Gene therapy holds great promise for treating a wide range of conditions, from inherited genetic diseases to various types of cancer. This is a rapidly evolving field with significant potential to revolutionize the way we approach and treat many medical conditions. The key is to precisely target and modify the specific genetic defects that underlie the disease, which can lead to more effective and personalized therapies. While still an emerging field, gene therapy is rapidly advancing and offers exciting possibilities for the future of medicine. The concept of gene therapy has been around since the 1960s, when scientists began exploring the possibility of replacing defective genes. The first human gene therapy clinical trial started in the 1980s, focusing on treating genetic diseases like severe combined immunodeficiency, SCID. The early 1990s saw some successes, but also setbacks, highlighting the complexity and challenges of gene therapy. The development of new gene delivery methods, such as viral vectors, improved the efficiency and safety of gene therapy. The late 1990s and early 2000s witnessed a surge in gene therapy research and clinical trials, expanding its potential applications. The 2010s marked a turning point with the approval of the first gene therapies, paving the way for a new era of therapeutic approaches. The goal of gene therapy is to treat or prevent disease by replacing, inactivating, or introducing new genes into cells. The first step is to identify the target gene, the gene that is causing or contributing to the disease. Next, we need to design a therapeutic gene, a healthy version of the defective gene, or a gene that can correct the disease. The therapeutic gene is then delivered to the target cells using a carrier or vector. Finally, the therapeutic gene integrates into the cell's DNA and starts producing the desired protein to treat the disease. Viral vectors are modified viruses that can efficiently deliver genetic material into cells. They can be engineered to target specific cell types, making them a powerful tool for gene therapy. Non-viral delivery methods include physical techniques like electroporation and gene guns, as well as chemical methods like liposomes and nanoparticles. These non-viral approaches provide alternative ways to introduce genetic material into cells. Viral vectors use viruses as delivery vehicles to introduce genetic material into target cells. Viruses naturally infect cells and insert their own genetic code, which scientists can leverage for therapeutic purposes. By removing harmful viral genes and inserting the desired therapeutic gene, modified viruses can safely deliver genetic material to target cells. Viral vectors can be administered through various routes, including intravenously, intramuscularly, or directly to the affected tissue. This allows for targeted delivery of the genetic payload to the appropriate cells or tissues. Liposomes are lipid-based nanoparticles that can encapsulate DNA and deliver it into target cells. Nanoparticles are designed to protect DNA from degradation and promote efficient cell entry. While direct injection of naked DNA can occur, this method is less efficient than other techniques like liposomes and nanoparticles. Electroporation uses electric pulses to create temporary pores in the cell membrane, allowing DNA to enter the cell. These four non-viral delivery methods each have their own advantages and can be used to introduce genetic material into cells. Gene replacement therapy aims to correct genetic disorders by introducing a functional copy of a missing or defective gene. Viral vectors, such as adeno-associated viruses, AAVs, are commonly used to deliver the therapeutic gene into target cells. Gene replacement therapy holds promise for treating a range of genetic diseases, including cystic fibrosis and hemophilia, as it has the potential to provide a cure. Gene silencing is a key approach in gene inhibition therapy, aiming to block the production of harmful proteins by silencing specific genes. This therapy can prevent or reduce the severity of genetic diseases by inhibiting the expression of disease-causing genes. Two main methods used in gene inhibition therapy are RNA interference, RNAi, and antisense oligonucleotides. RNAi uses small interfering RNA, siRNA, or microRNA, miRNA, to target and silence specific genes. Antisense oligonucleotides are short, synthetic DNA or RNA sequences that bind to target mRNA, preventing its translation into proteins. Both of these methods offer promising ways to treat genetic disorders by directly targeting the root cause, the expression of disease-causing genes. CRISPR-Cas9 is a revolutionary gene editing tool that allows us to precisely target and modify specific genes. This powerful technique holds immense potential for treating a wide range of genetic diseases by correcting the underlying faulty genes. Gene editing therapy has shown promising results in treating various conditions, from inherited diseases to cancer. By addressing the genetic root cause, gene editing offers the possibility of long-term or even permanent cures. The image on the right depicts the intricate interactions between CRISPR-Cas9 and DNA, highlighting the technology's precision and potential. Gene therapy offers a highly targeted approach, addressing the root cause of genetic diseases rather than just treating the symptoms. A single gene therapy treatment has the potential to provide long-lasting benefits, eliminating the need for frequent interventions or ongoing medication. For patients with genetic disorders that are currently incurable, gene therapy offers hope and the possibility of effective treatment. By addressing the underlying genetic defect, gene therapy can significantly improve the quality of life for patients, reducing the burden of their condition. The illustration on the slide shows a healthy human cell with a tiny, glowing gene therapy vector inside, conveying the idea of targeted, transformative treatment. Gene therapy holds great promise, but there are significant challenges that must be addressed. Precise delivery of genetic material to target cells is crucial for therapy to be effective and safe. Efficient delivery systems, avoiding off-target effects, and reaching cells with minimal damage are key challenges. The body's immune system can recognize the delivery vector or modified genes as foreign, leading to inflammation, rejection, or complications. Long-term consequences of gene therapy are still being studied, including potential insertional mutagenesis, immune dysregulation, and unexpected side effects. Ongoing research and monitoring are crucial to ensuring the long-term safety and efficacy of gene therapies. Gene therapy trials are a critical part of developing safe and effective treatments. They involve a multi-phase process to thoroughly evaluate the therapy. In Phase 1, the focus is on establishing the appropriate dosage and confirming the treatment is safe. Phase 2 then examines the therapy's efficacy and any potential side effects. Phase 3 trials are larger scale, confirming the treatment's effectiveness compared to standard care. Finally, Phase 4 looks at the long-term safety and effectiveness of the therapy. This step-by-step approach ensures gene therapies meet rigorous standards before being approved for wider use. Glibra was the first gene therapy approved by the FDA in 2012, used to treat lipoprotein lipase deficiency. Since then, several other gene therapies have been approved for various diseases like cancer, spinal muscular atrophy, and hemophilia. Approved gene therapies offer hope for patients with rare and severe genetic diseases, providing potential cures or long-term disease management. These treatments represent significant progress in the field of gene therapy and its ability to address unmet medical needs. Hemophilia is a genetic disorder that affects blood clotting, leading to prolonged bleeding even from minor injuries. Gene therapy offers a promising treatment by targeting the genetic defect responsible for hemophilia. The goal of gene therapy is to deliver a functional copy of the deficient gene to the patient cells, correcting the genetic defect and enabling the production of clotting factors. Clinical trials have shown promising results, with gene therapy demonstrating the potential to significantly improve the lives of patients with hemophilia by mitigating the risk of excessive bleeding. The slide illustrates the gene therapy process, showing a person receiving an injection of genetic material, the cell with a healthy gene for blood clotting factor, and a happy person without bleeding. Cystic fibrosis is a serious genetic disorder that impacts the lungs and digestive system. Gene therapy is an exciting new approach that aims to treat the underlying genetic cause of CF. Clinical trials are currently evaluating the safety and effectiveness of gene therapy for CF patients. The image shows a scientist carefully working in a lab, representing the precision and care required for this cutting-edge medical research. I'll provide an overview of how gene therapy works and the latest developments in using it to treat cystic fibrosis. Cancer is a complex disease driven by genetic changes that lead to uncontrolled cell growth and division. Gene therapy offers a promising approach to treat cancer by directly targeting the underlying genetic drivers. Gene replacement therapy can introduce healthy tumor suppressor genes to counteract the effects of mutated oncogenes. Gene inhibition therapy can silence the expression of cancer-promoting oncogenes. Emerging gene editing techniques like CRISPR, Cas9 allow for highly precise modifications of cancer-related genes. These gene-based therapies hold the potential to more effectively treat cancer while minimizing the harmful side effects of traditional treatments. Neurodegenerative diseases like Alzheimer's, Parkinson's, Huntington's, and ALS involve the progressive loss of neurons in the brain, leading to impaired functions. Gene therapy holds great promise as a treatment approach for these conditions. Gene therapy can be used to correct genetic defects, replace lost or damaged genes, or inhibit the expression of harmful genes. Researchers are actively investigating various gene therapy strategies for neurodegenerative diseases. This slide provides an overview of how gene therapy may be applied to treat these devastating neurological conditions. Patients need to fully understand the risks and benefits of gene therapy before deciding to undergo treatment. We must ensure they provide informed consent. Gene therapy should be accessible to all patients who need it, not just those who can afford it. We need to address issues of genetic equity and make sure treatment is available to people of all socioeconomic backgrounds. While gene therapy is intended to treat disease, there is potential for it to be used for enhancement purposes as well. We'll need to carefully consider the ethical implications of genetic enhancement. Editing genes that can be passed down to future generations raises serious ethical concerns. Germline editing is currently prohibited, and we must thoughtfully examine the long-term consequences before reconsidering this policy. Gene therapy is a highly regulated field, requiring oversight from bodies like the FDA and EMA to ensure safety and efficacy. These regulatory agencies have stringent protocols in place to closely monitor clinical trials for gene therapies. Detailed monitoring of patient outcomes and long-term safety is critical given the novel and potentially powerful nature of gene-based treatments. Rigorous clinical trial procedures help build confidence in the safety and effectiveness of gene therapies before they can be approved for wider use. Gene editing technologies like CRISPR, Cas9 are rapidly advancing, offering greater precision and efficiency for gene therapy. The future will bring personalized gene therapies tailored to individual genetic profiles and disease characteristics. Artificial intelligence is playing an increasing role in identifying therapeutic targets and developing gene therapy treatments. New delivery systems are being developed for direct in vivo gene delivery into specific tissues and organs, eliminating the need for ex vivo manipulation. Gene therapy holds immense potential for personalized medicine by offering targeted therapies tailored to individual genetic profiles. It allows for more precise interventions, optimizing treatment outcomes while minimizing side effects, leading to improved patient care. This approach acknowledges the unique genetic makeup of each patient and aims to personalize therapy based on specific genetic variations. Personalized medicine leverages our understanding of the human genome to deliver customized treatments that are more effective and have fewer adverse effects. By accounting for individual genetic differences, we can provide therapies that are better suited to each patient's unique biology, leading to better outcomes. Gene therapy and precision medicine are transforming healthcare by tailoring treatments to individual patients. Genetic profiling helps identify specific gene variations that can be targeted with precision treatments. Advanced gene editing techniques allow us to precisely modify genes, opening up new possibilities for treating genetic diseases. Integrating genetic and clinical data provides a more comprehensive understanding of each patient, enhancing personalized care and improving outcomes. These innovations are ushering in a new era of more effective, individualized healthcare. Combination therapies that combine gene therapy with other treatments can create synergistic effects, improving effectiveness and reducing side effects. Integrating gene therapy with established treatments like chemotherapy and immunotherapy allows for a comprehensive, multimodal approach to disease targeting. This strategic combination of different therapeutic modalities can lead to optimal patient outcomes. The synergistic effects and multimodal nature of these combination therapies are key advantages over single treatment approaches. Gene therapy is a rapidly evolving field, but it comes with significant challenges around cost and accessibility. The high costs of research, development, specialized facilities, and trained professionals make gene therapy an expensive treatment option. Insurance coverage for gene therapy varies widely, leaving many patients facing financial barriers to accessing these potentially life-changing treatments. However, there are ongoing efforts to address disparities in access and ensure that gene therapy benefits diverse populations and vulnerable communities. As this field continues to evolve, it will be crucial to find ways to make gene therapy more affordable and equitable for all who could benefit. Gene therapy is a cutting-edge healthcare technology that can be transformative, but many developing countries struggle to access it. There are significant socioeconomic disparities that create barriers to accessing gene therapy in the developing world. However, efforts are underway to increase access and bridge this gap, which is crucial for global health equity. Investing in research and development of gene therapy within developing countries is a key part of this effort to improve access and outcomes. Gene therapy is a promising but still emerging field, and there are several key barriers that have slowed its widespread adoption. One major barrier is the high cost of developing and producing gene therapies, which makes them inaccessible for many patients, especially those with rare genetic disorders. There are also ongoing safety and efficacy concerns, as gene therapy is a relatively new approach with unknown long-term side effects that make some people hesitant to try it. Effectively targeting and delivering the genetic material to the right cells and tissues remains a significant technical challenge that researchers are working to overcome. The regulatory approval process for gene therapies is very stringent and time-consuming, which can delay the development and availability of these treatments. Clinical trials for gene therapy have very specific inclusion and exclusion criteria based on the disease, treatment, and patient characteristics. Patients with severe forms of the targeted disease who have not responded to conventional therapies are often prioritized for gene therapy trials. Genetic testing is essential to identify suitable candidates and determine the specific genetic mutation involved. Comprehensive medical evaluations are conducted to assess the patient's overall health, medical history, and potential risks or complications. Patients receiving gene therapy undergo regular checkups to track their health and the effectiveness of the treatment. Monitoring helps identify any adverse effects, such as immune responses or unintended gene modifications. Long-term follow-up studies are essential to evaluate the long-term safety and efficacy of gene therapy. Data collected from patients helps researchers understand the long-term impact of gene therapy and refine treatment protocols. This allows us to ensure the safety and well-being of patients receiving this cutting-edge treatment. Gene therapy research is advancing on multiple fronts to improve safety and efficacy. Researchers are working to develop safer viral vectors like lentiviruses and adeno-associated viruses, as well as exploring non-viral delivery methods. Advances in gene editing are enabling more precise targeting of specific genes and cells, reducing off-target effects. Gene therapy is moving towards personalized medicine, where treatments are tailored to individual patients based on their genetics and disease. Gene therapy holds immense promise for treating a wide range of genetic diseases. However, continued research is essential to address current challenges and improve the efficacy and safety of these therapies. It's also crucial that we carefully consider the ethical implications and ensure equitable access to these potentially life-changing treatments. While the future is promising, we must remain vigilant and thoughtful as this field continues to evolve.